Okay, thank you very much, Akos. Nice talk. And then, this is Toshiro Butler from University of Nebraska-Lincoln, part of the organization committee. And then I'm working on plant metabolomics with conventional GCMS. That I don't have so much ideas about spatial metabolomics. So then I'm very excited to learn about this here. Okay, then I'm not really a specialist, but I can imagine that data analysis is one of the very big challenge of the spatial metabolomics. And our second speaker today is a specialist on it. And then Dr. Theodore Alexandrov is a team leader and the ERC investigator at the EMBL Heidelberg, Germany. And his research group is developed a lot of software tools for spatial metabolomics, and I'm excited to learn about it. Okay, Theodore, please start your presentation.
Cool. Thank you very much for the introduction. And thank you for inviting me here. So to highlight some of the work that we have performed over the number of years, in particular by developing and providing the community, you and the community, two software tools. One is METASPACE and another one is the recently developed SpaceM, where METASPACE is a tool for spatial metabolomics and SpaceM more of a tool for single cell metabolomics. By the way, can you hear me all right in terms of audio?
Yes.
All clear?
Yeah.
Good. Okay, so let me start. My name is Theodore. I'm again very happy in particular to represent here at least some part of the European community. And I work at EMBL, which is European Molecular Biology Laboratory, which is an international organization based in Heidelberg, Germany, but also has other sites throughout the Europe. And what our team at EMBL does, we develop tools to enable novel cutting-edge developments and answer biological questions. And for developments in single cell and spatial metabolomics, it really requires a team effort. So it really needs more than a village. And so to combine really the expertise and formulation of the questions in biology and omics, then to really figure out how to use mass spec. And in contrary to the previous talk by Akash, we are not really mass spec experts, we're more of mass spec users. But even to use mass spec, it's very sophisticated, challenging technology. So you really need to have devoted specialists knowing how it works, when it doesn't work, and so on. Our core expertise is actually how to make sense out of this data, in particular by developing and applying tools of computer science, computational biology. And also when we feel that there is something which can be useful to us and others, then we turn it into a software. And here definitely having professional software developers is absolutely instrumental for us. Over the past years, we have crystallized our interest, and now we're trying to support developments in the field of biology and medicine by developing tools and methods able to reveal, characterize, and shed light in general on metabolic cell state. We all know that this is very dynamic process and also reversible. What's very interesting process, which is influenced by DNA and cellular programs, but even more interesting, it is really also influenced and impacted by other cells. And we can think about cells which neighbors, tumor microenvironment, or definitely in plants, there is other tissues, or it can be interactions between cells of different organisms. And here we can think about host pathogen interactions. It is also environment in general, and here it's microenvironment, but also global environment, like pollution or definitely pesticides and everything else, which can affect not only the particular organism, but the whole ecosystem. And then also definitely in biology and medicine, there is strong interest to understand how therapy can affect or reverse the metabolic cell state. So here in general, spatial and single cell metabolomics, again, as was beautifully introduced by Akash, who works in this field already for a number of years. And by the way, Akash, I learned a lot of things about, and I was motivated very strongly by your initial experiments in single cell metabolomics to really also contribute to this field. So really nice to present in the same session. So here definitely single cell metabolomics, spatial single cell comes very handy, not only because it provides context and heterogeneity, but also because it provides readout closest to phenotype. And I think what's also important for all of us to understand that metabolomics, which is based on mass spectrometry, it is often the fastest and cheapest omics readout you can get. So it becomes very important for large scale experiments, and we're seeing it happening more and more. So this is our imaging mass spectrometry setup. It is a commercial system. It is just open here. So, but we didn't build it ourselves. We use a commercial system once again, which combines laser on top here, then atmospheric MALDI here. And here you see our scientific officer Prasad, who takes a tissue section here, places it on a glass slide, then puts it in this chamber, closes the lid. And then the process described earlier will happen where the laser fires at a location, which later become a pixel. And then the molecules get desorbed from this pixel, they get ionized, and they get counted, and the molecular composition of the pixel, it gets really quantified and represented here as mass spectrum. So, and you can get images here by assigning all the intensities for a particular molecule, or after doing metabolite identification, you can find which molecules are really hidden behind these images. The setup that we're having here, it's a pretty cutting edge commercial setup of 5 to 10 micrometer pixel size. It can detect over 100 molecules in one go. This is untargeted analysis, can detect metabolites, lipids, drugs. Normally we operate with about 5 protocols. Each of these protocols is targeted towards a particular class of molecules. And again, what brought me into this field about 11 years ago, or maybe already 12 years ago, is that this technology already back then, and now even more, generates a lot of data, because we have a lot of pixels, we have tens of thousands of dimensions or peaks measured per pixel, so it clearly generates a lot of data. And what was also very interesting for me as newcomer in this field back then, to witness what has happened over the past 20 or 10 years, is that imaging mass spectrometry became the major technology for spatial metabolomics. So our focus in this field was on addressing computational challenges. And here, if you're interested on our personal perspective, so please check out our review. So, and here definitely it starts from signal processing, visualization, statistical analysis. But by 2020, these challenges, they got addressed already with a number of community efforts and also commercial efforts. So over the past 5 years, we have focused on molecular identification, which is the problem of finding which molecules are encoded in this data. And I'll not go in detail, why is it a challenge? Why is it not a trivial problem? But what we have developed a solution to this problem, which is called METASPACE, which is an algorithm published earlier, but also free and open source software. Anyone can use it. It is supported by European funding and also by the National Institute of Health for us to really run it on the cloud and develop it further. Long story short, anyone can submit their data to this cloud platform. In about 10 minutes, they can run it through our metabolite annotation or identification engine, find which molecules are encoded in the data, but they also can share this data publicly with everyone, which creates this continuously populated and growing metabolite imaging knowledge base. And here I would like to give shout out to 3 software developers and data scientists who are continuously developing this platform: Lachlan, Lucas, and Sergey. So here a quick demo how it looks. If you go to metaspace2020.eu, you will see a lot of public data there, in particular in projects. So here I'm opening one project which contains data sets. Sorry, it's not planned this time. This is actually a whole body section of a mouse. And this data sets was submitted by 2 pharma companies, Servier from France and Genentech from the US. What you're looking at, it is a whole body section of a mouse. You see organs here. The head would be here. The liver would be here. And now I can go through all these molecules, which again were already found by METASPACE. And now I'm going just visualizing this molecule one by one and comparing the annotations and sharing these results with my collaborators and doing visualization and so on and so forth. So it's pretty useful tool, and for us it's really definitely great to see that it became a community platform. So over the past 3 years, we have processed more than 15,000 submissions, where one submission is one data set. what's even cooler than more than 30% of them became public. So now anyone can go there and use these data sets for their biology or method development. It is used by many labs, many users. And here also what's even cooler for us is to see finally that after a couple of years when it started growing, so there is more and more publications coming up, and there are already by now more than 100 publications where someone used it or shared their data through this platform. Here, I'd like to go shout out to champions of open data. These are those who actually shared most data publicly. And here you actually see PNL, and we have the PI, Chris Anderton here. It's also Imperial from the UK, UTHSA from Texas, Max Planck from Bremen, University of Copenhagen, University of Texas, Austin, University of Rennes from France, University of Melbourne, Gießen, and so on and so forth. So these are really those labs which transform the field by sharing their data and making it available. So I actually went, especially for this talk, I went and pulled information, how much data we have on plants. And although we don't have categories of kingdoms here, I pulled information on the plant database called BrackhamDB. So, and I found about more than 200 data sets on METASPACE annotated with this database. And you see that there is different parts, leaf, seed, stem, root, and so on, from different species like maize, brassica, and some soil microorganisms as well, corn, and so on. And here you see also the contributing labs. And it's really cool to see that the top two submitters or users of plant database, Chris and Young Jin Lee. So they are actually speakers in this symposium. So we definitely have a great representation of the leaders in this field in this symposium. So with this, I would like to wrap up the first part of my talk with the take-home messages that metabolism, for many, many reasons, not covered here. Some they'll cover it in the previous talk. Some will be probably in the next. Demands to be done in situ on the single cell level. Imaging mass spectrometry became the major technology. And also what we are contributing to this field is METASPACE as cloud platform for spatial metabolomics. With this, I would like to switch gears, and I would like to present another method and tool that we have developed, which called SpaceM. So which got just recently published. And this is a method for single cell metabolomics. In general, single cell technologies and metabolomics was nicely introduced by authors previously. Here, I'd like just to pull information from PubMed, just to strengthen the message that although single cell metabolomics is maybe the latest and the newest and the youngest of single cell omics, but it is developing as rapidly or even maybe faster than other single cell technologies, such as transcriptomics, proteomics, and so on. So it's definitely a very exciting field to be in. And we recently developed a method called SpaceM. The method was developed by Luca Rapes, PhD student in our team, and got recently published. And Luca, what he has developed is the method how to apply it to cells. So we do not have results for the plant cells, so everything I'll show will be for mammalian cells from now on. So when cells are deposited on the glass slide, we fix them to stop the metabolism. Then afterwards, we do another round of microscopy, first round of microscopy to find where the cells are, to find their phenotype, fluorescent phenotype. Then we do imaging mass spectrometry to get these pixelated images. We run it through METASPACE to find the molecules hidden behind these images. And then what Luca figured out is a simple but clever method how to overlay these two types of data with subcellular precision. And after introducing normalization, we can assign the normalized intensities to individual cells, resolving all this sometimes convoluted intensities coming from pixels. So, and now we have this data, what we call spatial molecular metrics, where for every cell we have metabolite intensities, we have fluorescence information, morphometric properties, how big is the cell, how it looks, and so on. And then also spatial features, which cells are next to each other. So now let me show you how this method can be applied to a biomedical problem. And here I'm demonstrating the most developed example, how we applied it to NASH, or non-alcoholic steatohepatitis, which is an intermediate stage upon transition from healthy liver towards liver cancer, which is characterized by pro-inflammatory cytokines, but often is induced by diet. Here we worked with the in vitro model hepatocytes developed by Matthias Heikenwälder from German Cancer Research Center and the PG student Mira Stadler. So, and here you can already appreciate accumulation of lipid droplets, where yellow is lipophilic staining, showing individual lipid droplets within the cell, and you can appreciate the single cell heterogeneity even between the neighboring cells in this isogenic system. So first we were wondering whether we can find lipids which contribute towards formation of lipid droplets. And here you see microscopy, you see individual cells indeed react to the stimuli by inflating with lipid droplets. And after we do our SpaceM analysis, this is UMAP technology. Again, repeating the previous speaker, this is multivariate analysis where two cells which have similar metabolic profiles, they're plotted next to each other. But what's cool compared to many other single cell technologies is that for every cell, we have also microscopy image. So now we can go back to microscopy and find what is the fluorescence, quantify it with this LD540 lipophilic staining, map it here. And now we see that this population of cells, which looks distinct, actually has high lipid accumulation. And this is clearly confirmed by also our markers here. And if we now zoom in, we do marker analysis, where for every lipid, one dot here is one lipid. So we quantify how much it gets accumulated in inflamed cells. This will be lipids which are really strongly upregulated in the inflamed cells. And we do enrichment analysis for these lipids. We find their properties, these are neutral lipids, we find their classes, we find their functions, and so on and so forth. So next, we embarked on more sophisticated study where we stimulated hepatocytes in 4 different manners, where this would be control, healthy. This would be just fatty acids or model of fatty liver disease. This is on top of this with pro-inflammatory cytokine. And this is actually where we apply an anti-inflammatory treatment, small molecule, which is inhibitor of NF-kappa B. And we already see that the cells look different. So let's now zoom into their molecular content. I'm showing you another UMAP plot where one dot is one single cell. We have a lot of cells and we have more than 700 metabolites and lipids measured and detected for every cell. And if we color code cells by the condition, we see that, yes, we see difference between unstimulated and stimulated cells. What is even more interesting, if we now break it into 3 clusters and we start looking into the cluster which corresponds mainly to hepatocytes with the strongest inflammation, we see that there are also yellow dots here, and blue as well. That means even among cells in this previous stage of inflammation, where they're just fatty acids along, some cells, they are already in this strongly inflamed state. And the same for treated hepatocytes. Although most responded, some cells, they actually didn't respond to the treatment. So it's really very interesting because it really breaks down the cells into distinct state based on their metabolic profiles. Wrapping up, so what we did afterwards. So, I mean, afterwards we did a really nice study, but PowerPoint is not calibrated. I'm opening right now. How am I doing with time? I'm almost there.
You have a couple more
Cool.
minutes.
Yeah, it's just the last slide. Mm-hmm. The slides are almost open. Sorry about this. Yeah, this was the last slide. Yep. Okay. Sharing with you. Cool. Okay, so the final slide. So what we did here is validation. So first of all, we found markers by finding the pseudo time trajectory from the unstimulated to the strongest stimulated cells. We plotted intensities of all metabolites for all 30,000 cells along this trajectory. Some of them, they get upregulated at the beginning, some of them get upregulated at the end. So, but what's even more interesting that we did in vivo validation, where here you see intensities of the markers across single cells from here. And here you see intensities from livers of mice, homogeneated livers of mice, bulk analysis. And you can appreciate how well the results, the markers that we see in the mouse model of NASH, how well they actually represent the intensity we found in this single cell model using single cell method. This is definitely very encouraging. And also 2 markers here and other markers here, they were reported as the biomarkers in the human cohorts. This shows the potential of using single cell analysis to find some biologically and hopefully even medically relevant information. This, I'm wrapping up my talk. Single cell revolution is happening, and single cell metabolomics is already contributing here. So we have developed method SpaceM. Right now, it can be applied to cell cultures, and it can help discriminate cell types, but also what's even more important and more challenging to really discover and characterize metabolic states. With this, I'd like to wrap up my talk. I would like to thank everyone who contributed to this tremendous work, in particular Luca and Sergio, all other team members, our collaborators at EMBL, our collaborators at German Cancer Research Center, other collaborators, and definitely funders. And thank you very much for your attention. Sorry for the hiccup, and thanks to organizers for inviting me here.
Thank you very much, Theodore. Actually, that you're perfectly on time. So I don't see any questions on the question and answer that anyone want to ask any questions.
So Theodore, I think you kind of, I know in the interest of time, didn't talk too much about some of the challenges with annotating the metabolites in METASPACE. Maybe it's given us a workshop and also educational endeavor, maybe just going through a couple high points about what the challenges with annotating metabolites are from mass spec data would be useful.
Okay. Yeah, so some of the challenges here is that the type of mass spectrometry we and most use for imaging is not exactly the type of mass spectrometry that is used for bulk metabolomics and lipidomics. So there are a variety of technical reasons for this, and but this requires to have some custom methods, and like METASPACE, but also there are other methods as well. And another challenge is that the results that we're getting, they can be ambiguous. So we should keep this in mind when we're reporting, when we're looking at the data. And here also definitely use some specific biological information which helps to do this. Or we try, for example, in METASPACE, when we show any results, we try to warn you if there is any sort of ambiguity. So in our experience, it is kind of like it requires either experience in mass spectrometry in your biological system, or we advise you to use some tools which are devoted to particular biological system, like, for example, plants.
Okay, thanks. We have one question from the audience so the Nandar Kumar, yes, sorry, how much you realize single cell metabolomics for plant tissues?
Yes, so unfortunately, like we don't do plant tissue as much. So again, so you probably have heard and have seen the talk by Akash previously, and will be definitely more in the future. Also today, and Young Jin Lee, I'm pretty sure I will be showing some results. We are supporting it from the computational perspective by adapting our tools, in particular METASPACE, so that others who run imaging mass spectrometry on plants, they can actually use it to find plant-specific metabolites. And also Chris Anderton, who is not presenting here, but also doing this work, and we're collaborating with him as well. But we do not have our original data to show because it lies outside of our direct area of like collaborators and availability of the systems that we have.
Yeah, that makes sense. So, and another question from the panelists in there, and the question is, how do you fix your cells before analysis?
Okay, yeah, so we fix it with a PFA 4% for 10 minutes, which is pretty standard fixation in cell biology. So what it does PE3 disrupts the membrane, gets inside, and cross-links the proteins, in particular metabolic enzymes, thus preventing enzymatic degradation. So what's also very important is that you keep into account that degradation can be not necessarily enzymatic only. So it can be oxidation, it can be other sorts of degradation. Here, also shout out to work done by Chris Anderton. They investigated that even when you store your sample in -80, over a long period of time, in particular, when not properly stored, you will have metabolic degradation. So this is another challenge of metabolomics per se, that you need to understand all these caveats and you need to plan it into your experimental design.
Right, that's very important. Yeah, and maybe last question that it's if you're analyzing the plant tissue, that what would be the best way that you can suggest for the cell fixation?
I feel bad that I cannot answer these questions because, again, we do not work with plant tissues, but I'm pretty sure that the speakers past me, they will be happy to take these questions because they have tons of experience working with plant tissues. Maybe I can forward it to them.
This will be a great topic for the panelist discussion.
Yeah. Okay, it's a time. So thank you very much, Theodore. Very nice.